Right, so you're probably aware of proteins, of carbohydrates. Probably the other class of molecules that we need to know about are lipids. Most of the lipids that you take in your diet are taken in as this kind of triglycerides, and the lipids that the fat that you store in your body is mostly stored as triglycerides. So the important energy storage molecule in animals and in plants as well. What plants store in their seeds, they'll have a lot of oil, which will be triglycerides stored there for when the seed germinates as an energy source. Lipids also, fats are important in animals as a kind of protective material around the essential organs. Anyway, so here is a triglyceride molecule shown here as a diagram. This thing, so it's made of a glycerol molecule drawn in green, and each glycerol is joined to 3 fatty acids. So if we actually, that's a fairly good way of looking at it, but we want to know what atoms are present in there. Then we look at this on the right. Now you can see that the glycerol part is drawn in green. Now that is glycerol is an alcohol. It contains OH groups. So if I draw a glycerol before it joins up with a fatty acid, it is just it's got 3 OHs on it. So we have 1 on each carbon. Don't forget, carbon forms 4 bonds, oxygen always forms 2, hydrogen 1. So that's glycerol, and it's an alcohol. We've come across one alcohol before at GCSE chemistry. Come across ethanol. So ethanol is different because it's only got 1 alcohol group on the molecule, but it's essentially the same kind of thing. Fatty acids. Now, fatty acids, we've also come across these sort of molecules in a way before, because fatty acids are simply, they are carboxylic acids. So we remember this, which is ethanoic acid, which is present in vinegar. So it's only 2 carbon atoms long, and it contains, this is the acid group. Okay, so you can see that these fatty acids are they are carboxylic acids, but instead of just being 1 carbon long, this is a great big long chain of carbons with hydrogen stuck onto it. And it's usually, fatty acids are always like 14, 16, 18, or 20 carbon atoms long. You never get them even numbers because they're made up, the way they're synthesized in cells, they're made up of 2 carbon units. Okay, so let's get rid of that bit there and have a look at how the fatty acids and the glycerol are joined together. Right, so right, now if you have a look here, this might be familiar, but I would be surprised if you can remember it. The way they're joined together is an ester linkage. So if I show you what an ester linkage is, an ester linkage is formed between an alcohol and a carboxylic acid, you may remember. So the ester linkage I've drawn in the purple brackets there. And there are 3 ester linkages in a triglyceride because you've got 3 fatty acids stuck on each one to each alcohol in the glycerol. Right, you just have a look how I've drawn this here. Now on the left hand side, this fatty acid here, I've drawn all of the bonds. So each carbon has got to form 4 bonds. So you can see most of the carbons, they are, like that one is from a carbon-carbon bond there, carbon-carbon, that means it's got room for 2 hydrogens. So usually that is drawn, rather than drawing like that, it makes it more convenient if you draw it as simply CH2. This one here, CH2, and so on. I've drawn a series of dotted lines there because I'm not going to draw all 18 of those because it takes too long. And when you get to the end, of course, the carbon has got to have 3 hydrogens because it's not forming to another carbon. So you'd write that one as CH3. Now, because that's pretty time consuming to write that, very often you use the skeletal method, which is the way I've drawn it here. So where in these zigzags, every one of those is a carbon atom. you don't put the C in because it's easier, and you don't draw the H's on it. You just presume that however many bonds are free, they are taken by hydrogen atoms. So all of those would have, because they're forming all these ones here, they're all forming 2 bonds to other carbons. That means they've got 2 bonds free for the hydrogen. So they're all CH2, except for the N1 there, that's going to be CH3. So that's a quick way of drawing it. So if you wanted to draw the whole thing, a fatty, the whole triacyl glycerol in skeletal form, you draw it like this structure over here. So you can see, there's a carbon atom on each of they're the carbon atoms of the glycerol, and so on. You've got carbon atoms on each of these zigzags. Right. So it's just a skeletal formula is just a much quicker way of showing it. Let's just show how the ester linkage is formed. Then I've only drawn. Oh, here's another point here. I've written down R. That's a very common way of, if you've got something, like a long chain of like CH2, 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 we just write it down as R. Says you write down the whole thing. That's how I've drawn that there. So we've got this is a fatty acid. What happens is you're going to lose water, as is usually the case in these. We've come across condensation reactions before. So you lose that water molecule, and the oxygen, of course, has got to form 2 bonds. So the oxygen is forming a new bond to that carbon, and the carbon is forming 4 bonds, and that's you've now formed the ester linkage, which is here. Okay. Most of the time, though, we're just going to be fairly okay. We're thinking of fatty acids. That's why we're thinking of triacyl glycerides being, the way I've represented it there. The glycerol and 3 fatty acids. When you're taking that into the body, you may remember from when you eat it, you may remember GCSE, you produce an enzyme called lipase, and lipase actually causes the breaking of the ester linkages. You've got to add water back, so it counts as hydrolysis. You break those and you get the fatty acids and the glycerol released from the triacyl glyceride, or just triglyceride, triacyl or triglyceride, you can call them. Okay, now let's have a think about fatty acids, because you may have heard of saturated fats, saturated fatty acids. That means there are no carbon-carbon double bonds in the molecule, in the fatty acid molecule. Unsaturated means you've got somewhere in the molecule, you've got at least 1 carbon-carbon double bond. So if I draw a fatty acid here, right, so here is the carboxylic acid end of it. Then I'm going to draw this as zigzags. Okay. I should really count 18 there. Okay, I'll count 18. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Right, I'm going to put a double bond somewhere in there to make it an unsaturated fatty acid. So I'm going to put one. I'm going to put one here. Okay, now I have put that double bond, I put it 3 from the end. So that was, if this is the end carbon, that's number 1, number 2, number 3. So it's between 3 and 4. And you'd call that an omega, which is a lowercase omegas like that. That's an omega-3 fatty acid. You may have heard of them. People go on about them being in ingredients in foods that's rich in omega-3 fatty acids. Good for you. So very often they're found in position 3. Another place where they're found is very often they're found at number 6, omega-6, and also omega-9. They tend to be found in those places rather than just randomly in the molecule. Now I'm going to draw, I think I'll draw this as a 9. Okay, so I'm going to get rid of that double bond there. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So I'll put a double bond in there. Okay, now if I put a double bond in there, something interesting happens is because this it produces a kink in the molecule. So I'm going to sort of get rid of that. All right, I'll rub it out. And you find that you get a kink is introduced into the molecule. Okay, I'm not sure if you're on the right number there. This always happens whenever you get a double bond. And this is called the cis configuration. If you do A-level chemistry, you will, the modern name for the cis configuration, you might have heard of it, is the Z form. Okay, but in biology, you normally call it the cis. And you may have heard like cis fatty acids, they're good for you. Now, the opposite of a cis fatty acid is a trans fatty acid. And a trans fatty acid, you don't have the kink in the molecule, it's more the straight. So if I drew it as trans, I'll draw it in red. The trans version would be like that. Trans versions don't exist naturally. They're sometimes introduced when foods are processed, and they are very bad for you. They cause an increase in blood cholesterol, much more so than just normal fatty acids, and they don't exist naturally. All unsaturated fatty acids are always in the cis configuration, which is the one there, and you've got a kink in it. And if you have a look at the phospholipids video, you'll see why it's very important sometimes that you have the cis configuration. It stops the fat molecules packing together closely, and that's really important. Before we finish up, let's just have a think of why do we call these fatty acids? Okay, so fatty in biochemistry and chemistry, fatty means there are no polar groups. It's non-polar. That means it is hydrophobic. It hates water. It won't mix with water. If you have polar groups, whenever you have polar groups, that's when you have things like oxygen atoms and nitrogen atoms, and they tend to make things water soluble. Now you can see triglycerides, they have got a few oxygen atoms up here, but most of it, they haven't got any at all. Most of the molecule is actually just this carbon and hydrogen, which will mean that it's hydrophobic and it will not mix with water. If you do chemistry, fatty groups, hydrophobic, basically means the kind of intermolecular forces that you're forming van der Waals forces. That's what it means. Okay, and when you have loads of carbons and hydrogens with no polar atoms, which cause molecules to be polar like oxygen, you can call it fatty, you can call it hydrophobic. It means it will not mix with water.